Today we are going to see about another interesting enzyme in the series of isoenzymes, that is nothing but HMG CoA synthase. So let's get into the contents. HMG CoA synthase is also called as 3-hydroxy-3-methyl glutaryl CoA synthase. Has a EC number of 2.3.3.10. It is a key enzyme that catalyzes the condensation of acetyl CoA and acetoacetyl CoA, and we have formation of HMG CoA, that is 3-hydroxy-3-methyl glutaryl CoA. It exists as two isoenzymes with distinct cellular localization, function and metabolic roles. So let us see about the two isoenzymes of HMG CoA synthase in detail. The first form is a cytosolic HMG CoA synthase. It is distributed in tissues such as liver, adrenal cortex and gonad. It works on lipogenesis and sterol biosynthetic pathway. It is involved in cholesterol biosynthesis in mevalonate pathway, and it supplies HMG CoA for conversion into mevalonate by HMG CoA reductase, which is a rate limiting step of your cholesterol biosynthesis. So this is the role of cytosolic form of HMG CoA synthase. In the fed state, in the presence of insulin, glucose is converted into pyruvate. Pyruvate is further converted into acetyl CoA in the cytosol. This acetyl CoA is converted into acetoacetyl CoA by the enzyme thiolase, and acetoacetyl CoA and acetyl CoA in the presence of HMG CoA synthase leads to the formation of HMG CoA. This HMG CoA is reduced into mevalonate by HMG CoA reductase, and this mevalonate leads for the formation of isoprenoid units, which leads for squalene synthesis and finally ends up in cholesterol biosynthesis. And cholesterol acts as a precursor for various steroid hormones, bile acids and vitamin D. So the pathway association is conversion of acetyl CoA and acetoacetyl CoA into HMG CoA. We have mevalonate, cholesterol and various steroid hormones, bile acids and vitamin D production because of the presence of this cytosolic form of enzyme. Coming to the regulation, it is upregulated by sterol regulatory element binding protein, and it is suppressed when cellular cholesterol concentration is high and insulin stimulates its expression. Coming to the another isoform, which is mitochondrial HMG CoA synthase. It is located in the mitochondrial matrix of the liver. It catalyzes the first committed step in ketogenesis, that is, it produces HMG CoA used for ketone body formation. It is encoded by the gene HMG CS2 gene, and it is essential during fasting, starvation, diabetes mellitus, low carbohydrate diet. Coming to the pathway association, it is involved in beta oxidation of fatty acids. Finally, we have production of acetyl CoA, acetoacetyl CoA, and from acetyl CoA and acetoacetyl CoA we have formation of your acetoacetate and beta hydroxybutyrate, which further leads for the ketone bodies in the blood system. So this is the pathway. During fasting and starvation, when the glucagon levels are high, in adipose tissue we have lipolysis, which leads to the fatty acids, and this fatty acids enters liver, and in mitochondria of the hepatocytes, beta oxidation takes place and acetyl CoA is produced. Two molecules of acetyl CoA is converted into acetoacetyl CoA, and acetoacetyl CoA combines with acetyl CoA to form HMG CoA, and HMG CoA lyase converts HMG CoA into acetoacetate. Further, we have ketone bodies which are released into the bloodstream, and it is converted back into acetyl CoA, where acetyl CoA enters the TCA cycle and leads to the formation of ATP. This mitochondrial form is regulated by hormones such as glucagon, and it is induced by fasting state and PPAR activation, and it is inhibited by insulin. Coming to the significance of both the forms, overactivity of cytosolic form increases cholesterol biosynthesis and leads to cardiovascular risk. Target indirectly affects by statins, which inhibit HMG CoA reductase, which is downstream the pathway. When coming to the mitochondrial form, deficiency leads to impaired ketogenesis, hypoglycemic in infants, and inability to adapt to fasting. Thank you for your patient listening. Let's come back with another isoenzyme and with interesting topic ahead. Thank you.